Welcome, everyone. We will just wait for everyone to join the meeting and we will start shortly. Welcome, everyone, to this last ESDR kitchen of the year. So my name is Katia Boniface from the ESDR, and I have the pleasure to chair this session with Katrina Abuabara from the SID and Akiharu Kubo from the JSID. And today's topic is about environmental influences on dermatologic disease. So it is an ISID flavors episode, which aims to bring together the societies for dermatological research from US, Asia, and Europe. And today we have two great speakers that will share their research. So Yumi Nakamura and Ian Myles. And so I will let Akiharu introduce Dr. Nakamura.
Okay, so hello everyone, and this is Akiharu Kubo from Kobe, Japan, and I'll chair the first part of today's ISID flavors. And the first speaker is Dr. Yumi Nakamura, and she is now professor of Osaka University, Japan. So she is long-term achieved very fantastic works of skin microbiota and the environment of our skin surface. So please, Yumi, start.
Thank you, Akiharu, for your kind introduction. Hello, everyone. I'm Yumi Matsuoka. Thank you for joining. Let me share my slide. Can you see my slides? Okay. So today, of course, most of you may know in atopic dermatitis, pathogenesis, skin barrier defect, and Th2 immunity are most important factors. But according to today's topic, in the next 10 minutes, I'll introduce key aspects of axial column sensing in staph aureus and how this communication system relates to the skin microbiome and the atopic dermatitis. So I'd like to start by explaining dysbiosis in the skin microbiome associated with atopic dermatitis. Recently, there has been growing interest in the skin microbiome and its role in various skin diseases. However, the phenomena of dysbiosis, where the skin microbiome in atopic dermatitis differs from that of healthy skin, has been known since as early as 1960s and 1970s. This paper, actually published in JID in 1976, investigated skin bacteria in atopic dermatitis and healthy individuals using culture-based method. It reported that specific colonization of staph aureus in atopic dermatitis. I was particularly impressed by how the authors refers to the skin microbiome as an ecosystem in the title. In short, the discovery of dysbiosis in atopic dermatitis skin due to staph aureus colonization is not something entirely new. In the 2010s, the development of next-generation sequencing revolutionized microbiome analysis, bringing renewed attention to dysbiosis in various diseases. Here I'm showing a well-known microbiome analysis data published by the NIH group in 2012. As you can see, the proportion of staph aureus increases significantly during atopic dermatitis disease flare, where this method clearly shows the rise of staph aureus in lesional skin, but it does not explain how staph aureus contributes to the pathogenesis of atopic dermatitis. To address this, we focused on understanding how staph aureus induce dermatitis through the activation of its AGR quorum sensing system. So next, I would like to introduce about bacterial quorum sensing. As we humans, we can recognize congestion with our own eyes. When we are in the private pool, we feel very relaxed like this, but in a crowded space like this, we feel stressed and find it hard to interact with others. Similarly, bacteria secrete pheromone-like peptides to sense their density. Quorum sensing is a cell density-dependent signal transduction system that controls a variety of physiological behaviors in bacteria and their survival. These responses include adaptation to nutrient availability, defense against other microorganisms that may compete for the same nutrient, and avoidance to toxic compound potentially dangerous for the bacteria. For the control virulence, staph aureus employs AGR quorum sensing system. The AGR operon is organized around two divergent promoters, P2 and P3. These two promoters, P2 and P3, work together to regulate AGR gene expressions. Here you can see a peptide pheromone called OAT-inducing peptide. AIP is translated from AGRD region. AGRB transport AIP into extracellular space and AIP bind to the extracellular domain of AGRC receptor. After that, AGRA and C form a two-component signal transduction system and positively regulated the AGR expression itself. And this also regulates downstream signaling. RNase3, which encodes delta toxin, also works as a non-coding RNA that controls several downstream virus gene expressions. Not only delta toxin, but also PSM alpha that I will talk later, both are strictly regulated by this system. Our research team has been studying how staph aureus proliferates on the skin and contribute to the pathogenesis of atopic dermatitis. On the skin where staph aureus grows and its AGR quorum sensing system is once activated, as shown here, downstream PSM alpha PSM alpha production damage epithelial keratinocyte. This damage leads the release of alarmins such as IL-1 and IL-36, which triggers IL-17 dominated inflammation. Additionally, another downstream product of AGR quorum sensing, delta toxin, directly acts on mast cell, induce Th2 type skin inflammation. Moreover, a recent study by another research group reported that the quorum sensing downstream V8 protease directly stimulate the PR1 receptor on each sensory neurons. This not only induces itself, but also promotes scratching behavior, causing further skin damage. First, as I mentioned earlier, the AGR quorum sensing, which plays a role in inducing dermatitis, is also involved in the colonization of staph aureus on the skin. Through clinical sample analysis, we have identified that this mechanism is associated with the onset of atopic dermatitis during infancy. In this study, we performed whole genome analysis of staph aureus isolated during the preclinical phase from two group infants who developed atopic dermatitis by 1 year old and healthy infants. By comparing the whole genomic regions of these clinical isolated staph aureus strains, we investigated whether any factor regulates staph aureus colonization before the onset of atopic dermatitis. Interestingly, we discovered that only the strains isolated from the skin of healthy infants developed loss of function mutation in the genomic region of AGR quorum sensing. And this phenomena did not happen in atopic dermatitis associated infant skin. The right panel, the result of mouse experiment conducted to confirm whether the loss of AGR quorum sensing function affects skin colonization. When wild-type staph aureus was applied epicutaneously, severe dermatitis developed in mice like this with the extensive green-stained bacterial presence in immunohistochemical staining. In contrast, AGR quorum sensing deficient strains did not colonize the skin, nor did they induce inflammation. These results demonstrate that the AGR column sensing of Staph aureus is a key driver of skin colonization and the development of atopic dermatitis. At this point, based on the findings I've shown so far, I think a natural question arises. Could AGR column sensing itself serve as a therapeutic target in atopic dermatitis? In the following few slides, I'll discuss what current evidence suggests. This slide illustrates how commensal and virulent staphylococci interact through the AGR column sensing. The skin commensal staphylococci, such as Staph epidermidis or Staph lugdunensis, also utilize AGR column sensing. As shown here, the auto-inducing peptide sequence of these commensal staphylococci differ from those of Staph aureus. Interestingly, commensal AIP have been reported to inhibit AGR expression in virulent Staph aureus. This mutual inhibition is thought to play a critical role in bacteria competition in the environment. Interestingly, a previous paper also demonstrated that if the atopic dermatitis patients have less commensal AIP in the skin, they have more severe skin symptoms. This graph shows that the amount of Staph epidermidis AIP in the skin of healthy, less severe AD and severe AD. You can see more severe AD patients have less epidermidis AIP in the skin compared to those of healthy and less severe AD. Then some previous papers also demonstrated that the commensal AIP inhibits Staph aureus skin colonization in the mouse models and atopic dermatitis patient skin. This study demonstrated that mice infected Staph aureus intradermally with commensal staphylococci or commensal AIP showed reduced skin ulceration. The bottom study is a phase 1 clinical trial in which atopic dermatitis patients were treated by topical commensal bacteria. Commensal Staphylococcus bacteria. You can see the suppression of AGR column sensing of Staph aureus by commensal staphylococci slightly improve the local skin inflammation in atopic dermatitis patients. To date, unfortunately, this approach has not shown clear efficacy in larger, well-controlled clinical studies, and these kind of therapy still need to be improved, I think, but the negative regulation of AGR column sensing is a promising therapeutic target of Staph aureus-induced skin diseases. This is a conclusion slide. We previously demonstrated that increased colonization of Staph aureus induced PSM production through AGR column sensing activation, and these toxins led to barrier disruption and Th2 immunity. Additionally, AGR column sensing seems to be required for successful colonization on infant skin and contribute to the onset of atopic dermatitis. Today, I did not introduce the study, but our recent another infant cohort study has also related several key findings about the skin commensal bacteria regarding atopic dermatitis development in the neonatal period. That's all of my today's talk. Thank you for listening. I'm happy to take any questions.
Okay, thank you, Yumi. And we will take questions after both speakers finished the talk. So, Katrina, please.
Yes, thank you for that wonderful talk. I'm now going to introduce Dr. Ian Myles. He's currently Chief of Epithelial Therapeutics at the National Institute of Allergy and Infectious Diseases in the U.S. He's a tenure-track investigator, and he's recognized by the prestigious Lasker Clinical Research Scholar and Distinguished Scholars Programs. He has not only done academic work, but in 2011, he became the commissioned officer in the United States Public Health Service Commission Corps, and he supported several missions, including development of the Ebola virus vaccine in West Africa. He's published over 70 high-impact articles and leads a very active clinical and translational research program, and I'm really excited to hear his talk today. Thank you for being here with us.
Okay. Thank you guys for having me. Let's see. All right, should be good. Okay, so if one were to graph out the incidence of atopic dermatitis on a historical scale, it would look something like this. The very first description, obviously they did not use the word eczema, but the first description of an itch that rashes and the same kind of dermatologic features was on the Ebers papyrus, pulled out of a pyramid. Hippocrates talked about eczema. Emperor Augustus had the allergic triad. But it's really not until the 1970s where we see this explosion of the incidence. And you can contrast that with, I know I'm not getting into it, but there's debates around certain neurodevelopmental disorders about whether their incidence is genuinely going up or recognition. For atopic derm, it's pretty clear that it's genuinely going up, and particularly for food allergies. And yet, so that tells you something in the environment had to have changed in the 1970s. And I invite you to Google this for yourself, just in case my algorithm is feeding me biased pictures. But they will all look something like this, where the top of the image is the skin and everything beneath it is kind of the biochemistry, something along this line where maybe there's a hand, and now, like the previous speaker introduced Staph aureus, but not really much else in the environmental context. And this has been great because these are the targeted molecules for which a lot of these drugs are created. But when I see this, I see an image that looks something like this, where you have mechanical disruption, electrical abnormalities, and some things in this picture are literally inflamed. But if you can study this as much as you'd like, it will never tell you how things started. What was the very first thing that went wrong that then has led to all of this? And so kind of the first set of studies, we were happy to be part of one of them, came out in 2023. So 4 different studies, 4 different countries, using 3 different modalities between mass spec, shotgun metagenomics, and RNA-seq. Each of these studies have their own claims of kind of what predates atopic derm. So what they did in all of these was collect a sample in the very first week of life, sometimes in the first few days, for children who did not appear to have any skin disease, and then just followed to see who developed atopic derm and who did not. And so to go back to say what was predictive of who eventually developed the disease. And again, each one had their own findings, but all of them came away with a ceramide as well as sphingolipid and phospholipid metabolism abnormalities were what predicted who was going to go on to develop atopic derm or not. So this becomes very intriguing for the microbiome. Again, it was mentioned the NIH researchers, Dr. Segre and Dr. Kong, had kind of mapped out where on the body you can expect certain organisms to be. And I have kind of colored in for them where the gram-negative organisms would be found. And the reason for that is when you go and look at all the different types of organisms in the universe that can make sphingolipids, phospholipids, and ceramides from a microbial standpoint, almost all of them down here are gram-negative organisms. The very few gram-positive organisms that are capable of making sphingolipids are also skin commensals. So that's kind of speaking to the fact that, all right, a lot of your bacteria on your skin can make these lipids. The few that don't make these lipids, like Staph epi, actually make sphingomyelinases, which enhance host production of ceramides and sphingolipids. So this is speaking to the possibility that, boy, it sure looks like the skin microbiome is providing the very things that predict whether or not you go on to develop atopic dermatitis. And so I threw this slide in at the last second just to say that if this is true, in theory, you could replace the organisms producing these bacteria. And this is currently still under an investigation for us, but also sold over the counter through NIH agreements. But I won't harp on that because irrespective of what's wrong with the microbiome, it does not answer the question, what went wrong in the 1970s? It's not, there's not an acute onset global dysbiosis for no reason. And so what has been shown in the literature of environmental factors associated with atopic derm can kind of fit, I think, in 4 flavors. The first is just living in an industrialized area, consumption of processed foods and antibiotics. This is almost certainly more relevant to gut microbiome and gut health. You can see maternal atopy matters. We've seen older siblings are protective, but another one is moving from a non-industrialized country to the United States or the EU greatly increases your risk. If you're under 2, and really the cutoff is under 4, after 6 years of age, you can move anywhere you want in the world, and your risk of allergic disease is not going to change from where your home country was. So this kind of speaks to some kind of early developmental window. Within the United States, there is an additional compounded risk if you live in an urbanized environment versus rural. Typically, when you use the word urbanized environment in an environmental setting, you're almost certainly talking about the second group, which is car exhaust. But if you look down kind of the major air quality index measurements, when you see that on the news, it's almost always measurements of some of these components. The associations are pretty weak at best. Some are non-significant, but there's a real big risk, and this has been shown in multiple countries now, of living near a highway, particularly if you're pregnant. If you're very close to a major road source, the chances that your child is going to have eczema is dramatically higher. Wildfires are a newly appreciated one, particularly after Australia and San Francisco. And then the last set is kind of what I would say a hodgepodge of home chemicals and home exposures, like wearing nylon, spandex, and polyester. Most of our patients gravitate away from those. Environmental tobacco smoke always makes things worse. And then some very strange ones like new flooring and furniture. Again, renovating your kid's nursery basically before the age of 2 confers a big risk, particularly if you wallpaper it. After age 4, decorate all you want, doesn't matter. So those don't really provide any real consistent signal. So what we wanted to do is say, well, can we make any sense of this? And in short, we're doing geospatial analysis to say, we took a visitation database to say where in the country seems to have a lot of eczema visits or atopic derm visits and where it has very little, and what are the chemicals that are most prominent in those areas from the Environmental Protection Agency. And just jumping through a lot of math, it's basically diisocyanates and xylene, which are related into a group that's called BTEX, or benzene, toluene, ethylbenzene, xylene. So this class of chemicals are the ones that show up geospatial analysis. You'd want to ask then, all right, well, does that make any sense? How would you be exposed to these various chemicals? Well, their manufacturing began in the United States in earnest right around 1970. This is for toluene diisocyanate. This is for xylene production put into polyester or nylon in the case of benzene. Before 1975, 0 cars produced either one of these chemicals, but as a side effect of the catalytic converter, every automobile after 1975 becomes a BTEX producer. The only natural source of these are timber waste from wildfires. And then if you wanted to fill your home with this, you would do non-latex foam mattresses, like in new furniture. You would wallpaper the house. You would smoke cigarettes. Again, the synthetic fabrics are all made directly from these chemicals. So there's an epidemiologic overlap for sure. Putting these in the laboratory, they can directly activate itch receptors like TRPA1. In terms of what they do to the bacteria, in brief, Roseomonas breathes in nitrogen and breathes out phosphatidylcholine to make into our ceramides, but we can shut that off. This is in red. That as I increase the dose of toluene diisocyanate, I'm reducing the overall ceramide production in our bacteria, even at concentrations that increase the growth. So Roseomonas can use this as a substrate, but in order to do so, it has to shift its physiology away from these lipids. And you can see this in the real world. So we took various pillowcases made from different fabrics and sprayed them with either Staph aureus or our good guy Roseomonas. And you can see at 20 days, Staph aureus does not grow well on natural components, but will hang out just fine. I don't know about how to wash that off yet, but yeah, Roseomonas, you might think, oh, this is great. It grows on all the different ones. But again, in order to grow on nylon or spandex or polyester, it has to change its physiology and it'll actually reduce its lipid output in order to survive. So it'll kind of turn a good guy bad. And so this has a lot of implications, obviously, for treatment, like I said, in terms of our topical disparities, in terms of which groups have a lot of exposure to these different chemicals, actual prevention, rather than saying we're going to slather a kid with some product instead, say, how do we remove the exposure that might be causing this? And many other diseases, including those in dermatology. So I'll just, for 1 minute, mention we created this entire website, which we're calling Paddle, which will allow you to look through every single disease in the same way that we did. So we can look at something by chemical and say, how associated is this chemical with every individual different disease? Where does this chemical show up in the United States? How strongly does it associate with poverty and segregation, basically? And then you could go the other direction and say, all right, I want to know what chemicals are associated with the disease that I'm interested in for children and adults. what kinds of chemicals those are and what biochemical pathways do those chemicals influence. And so our idea, these are obviously associations, but the idea is that they would then lead to mechanistic studies as well. And so with that, I'm out of time. So we'll just say we'll work to do more. Thank you.
Thank you so much. That was a really wonderful tour of history and science. So now we'll open it to questions. If anyone has questions, please put them in the chat and we can share them with the speakers.
Maybe I will start with the first question during the time people are writing their questions. So thank you to both of you for these really nice talks. So maybe a first question to Dr. Myles. So because you nicely showed that the risk is increased before the age of 4 years old, basically. So I was wondering whether there was a link established with the acquisition of the immune system, because we know that from birth, so like babies don't have any immune system, and usually it's like we say between like the 6 to 8 first years that we are exposed to a lot of environmental like triggers, and that we are establishing the immune system. And I was wondering whether this was linked, this increase linked to that, or if it was like completely independent.
I don't know that anyone's directly linked it. I mean, our hypothesis would be that if you started out your life effectively deficient in these lipids because your microbes aren't providing them for you, then you're going to have all the downstream problems. Your barrier function is not going to be as strong, so more things are going to get in, generating more immune activation. You're not going to have the competitors like Dr. Nakamura was showing Staph aureus. Like you want to have something that would take up that space, so then all the bad things that staph could do, you're now susceptible to. So that's how kind of we do it. But again, there has to be some kind of developmental window because people who again are in their 30s who move to the United States from rural Uganda do not suddenly develop atopic derm. So there's some kind of susceptibility window that they're not falling within.
Thank you. Maybe I will ask now a question to Dr. Nakamura. So it's about, because something that is always intriguing me, so I'm more on the immune side of the, like in skin inflammatory diseases. And what we see, for example, in atopic dermatitis is that if you compare the lesional skin with like non-lesional skin of patients, that you already have some like of immune abnormalities, but still the patient like has not yet developed any like lesion. And I was wondering with your AGR quorum sensing, whether you could see some differences between the non-lesional skin and lesional skin of patients, if you looked at that.
Well, thank you for your important question. When we look at the compare the axial quorum sensing expression between the non-regional and regional, only regional skin we can see the axial activation. But we can still can see the colonization of the sufferers in non-regional. So maybe the bacterial density is very important. So regional skin more have more bacteria grow, and the density is very crowded, and it AGR only activated in the regional skin.
Okay. And did you look, so because you're showing as a therapeutic avenue with this AGR quorum sensing, so did you look how you are modifying the immune response?
Immune response, AGR
Yeah.
activation.
Yeah. So because you showed on one side that you are triggering IL-17, but then also on the other side you have IL-4 and IL-13. So I was wondering whether if you would modulate this quorum sensing activation, like by dampening or like activating it more, whether you would change the outcome of the immune response.
Yes, of course. If we can suppress the axial quorum sensing activation, we can suppress the Th2 immune response and the Th17 immune response. But in the development phase of atopic dermatitis. But after the atopic dermatitis development, we cannot see the difference between the non-regional and regional by bacterial colonization.
I have a question for Dr. Myles. I'm curious if you've thought a little bit about eczema or atopic dermatitis in older adults. We all know that eczema is really heterogeneous, but have you had a chance to think about the role of the microbiome or of chemicals or the ceramides and lipids in that population, and whether that may explain some of the trends we're seeing with increasing rates there as well?
Yeah, we have the chemical associations aren't any different between pediatric and adults. So these same BTX compounds within the United States are associated. I don't have access to or haven't seen kind of epidemiology evidence that would say like, all right, well, what would predict adult onset AD? Because we all know that it's not like everybody's in their 70s or something where you could attribute skin thinning or something like that. So I feel like I would need better ways to target to say, because the databases we have don't have good age. So it'd be nice to be able to say, like, are there places where adults are developing it in their 30s or 40s who didn't have it before, and then, down the line. So honestly, that might be something that's better for a system like the UK or one of these other countries that have, lifelong data in a centralized system. But I have thought about it. It's hard because the greatest exposure to these chemicals is actually like spray foam insulation or repolishing your floors to the benefit of the industry. I guess most people are not allowed to do spray foam insulation without the full zip up and the hat and the mask and all this stuff. So that might explain why they're not having problems, but you don't hear people in the factories suddenly developing it. So again, it can't be just I'm now moved to a polluted area and suddenly I'm developing eczema. I feel like that would have shown up. So yeah, I don't have a great answer for adult onset AD yet.
So
Fair enough.
Okay. So there's a question from the audience. So maybe this is to Dr. Myles. So do we know anything about the influence of different preservatives on the impact on skin microbiome?
Some. So we have a paper, it's kind of old now, I guess, but Carlos Castillo is the first author. And what we did was basically like what you would do for a penicillin assay, where you just grow it on a plate and plop the patch test on the into the center of it and measure how much of the inhibition it is, where you do sensitive and resistant. And so we tested all the ones that are in your standard patch test one. And then we went to the store and we bought every single thing that used the word eczema on the label to see. And it is pretty disturbing how some of these products will obliterate all the healthy commensals that we would grow on a plate, and Staph aureus is completely impervious to it. But of course, that's an incredibly biased sample because we're only picking things that would potentially cause contact dermatitis. All the different preservatives out there that have not been slapped onto a patch test, we don't really have genuine access to each of those. I thought there was some private company that was throwing out the idea that they were going to test these things and get some kind of seal of approval, but I haven't heard anything from them in a while, so I don't know how they did. But I mean, definitionally, a preservative is something that's trying to prevent bacterial growth in a product. And so from the product's perspective, it's a prophylactic antibiotic. So obviously it's going to have some kind of antimicrobial effect. It's just whether or not it does in our particular organisms. I would think that it would, and that would, whether or not the dose is high enough or whatever. But yeah, so there are some published, like I said, and conceptually you'd have to expect that there'd be a lot more out there.
Thank you. Oh,
So I think we have come to the end of this webinar. So again, I would like to thank you both, Dr. Myles and Nakamura, for joining us today and so sharing some of your research. So before the end of the meeting, so just a reminder to tell your colleagues who were not able to attend that the ESDR Kitchen webinars so will be an hour on YouTube. So for this one, it will be so in 1 or 2 days. And so it was the last episode of the year, but we will have, so in 2026, our first recipe book episode, so on January 14th. So it's back to the regular time, which is 1:30 in the afternoon, and it will be about genome editing in dermatology. And so last, I would like on behalf of the ESDR board to thank really all our chairs and speakers for this year's program, which was really rich in topics and with great speakers. And also thank you to the audience for your engagement and participation. And so we will see you next year. And so also, so we now can introduce properly. So our next ESDR annual meeting, which will be in Germany, in Heidelberg. So from September 9 to 12, so in 2026. So thank you very much to all. And so I hope to see you next year. Goodbye.